नमस्ते हामी चाहिँ एक्सपेरिमेन्टल एपिडेमियोलोजीमा चाहिँ प्रवेश गरेका छौँ र एक्सपेरिमेन्टल एपिडेमियोलोजीमा म वाचन गर्न गइरहेको छु ऱ्यान्डमाइजेसन कन्ट्रोल ट्रायल आरसिटी युनिट अफ स्टडी आरसिटी इज पेसेन्ट आरसिटी इज टू टाइप हुन्छ एउटा कन्कुरेन्ट प्यारलल डिजाइन कम्पेरिजन आर बिट्विन द टू ग्रुप हुन्छ भने एक्सपेरिमेन्टल ग्रुप इज एक्सपोज टु स्पेसिफिक मेडिकेसन अर इन्टरभेन्सन रिफरेन्स ग्रुप इज नट एक्सपोज टु स्पेसिफिक मेडिकेसन अर इन्टरभेन्सन crossover design comparison are made between two groups. Experimental group is exposed to specific medication or intervention. Reference group is not exposed to specific group, now become non-exposed and vice versa. यहाँले हेर्न सक्नुहुन्छ रेफरेन्स ग्रुपमा चाहिँ चेन्जेस छ भन्ने कुरा क्रसओभर डिजाइन आरसिटी हेल्प रिमुभिङ इथिकल कन्सर्न चाहिँ रिमुभ गर्न चाहिँ क्रसओभर डिजाइनले चाहिँ हेल्प गर्छ इन्टेन्सन टु ट्रिट ट्रायल इम्प्लाइज द्याट द रिजल्ट अफ आरसिटी आर अनएफेक्टेड बाई एट्रिएसन लस टु फलो अप अर चेन्ज ओभर अफ द स्टडी सब्जेक्ट फ्रम वान ग्रुप टु अनदर randomization in RCT. Randomization is a randomized control trial is a statistical procedure by which participants are allocated into either of two group experimental group in which the intervention is given and reference group in which the intervention is not given. Randomization is based on by random number table. The essential purpose of randomization is randomized control trial. Participants have equal and known chances of falling into either experimental group or reference group. To eliminate selection bias, to have similar prognostic factor among two groups, randomization remove both confounding and bias. दुईटै remove गर्छ. Randomization is superior to matching. Randomization ensure both known and unknown confounding factors are distributed equally among the two group, thereby nullifying their effect on result, whereas matching is useful for only non-confounding factor. Pre and post clinical trial. Pre-post clinical trial does not have a true control group. Patient acts as his or her own control. Each patient has a pre-test score followed by a post-test difference in score reflect change in attribution to intervention. Is often used in assessing whether knowledge, attitude, or pre-existing risk behavior changes when a subject is assigned to an intervention. Limitation: difficult to assess if change is due to developmental intercourse. Difficult to assess if change is due to regression, worsening to mean. Not useful in study involving mortality is post-test won't be available. More difficult to interpret than the comparable parallel clinical trial. Cannot be randomized. Preclinical and clinical trial phase unit of study propose preclinical phase lab experiment animal purpose चाहिँ pre-testing clinical phase मा चाहिँ phase 0 healthy human volunteer micro dosing phase first healthy human volunteer safety and non toxicity profile phase second patient efficacy को लागि गरिन्छ Phase 3 patient comparison with the existing drug, whereas phase 4 patient long-term and rare side effect को लागि गरिन्छ. Phase 3 is an RCT comparison of new drug with existing old drug. New drug is launched in market after phase 3. Longest phase of trial is phase 4. Post-marketing surveillance is phase 4. Maximum tolerated dose of drug is phase 1. Maximum drug failure reported is phase 2. Meta-analysis description. An objective systematic review that employs statistical method to combine and summarize the result of several studies. A quantitative synthesis of all unbiased evidence meant for summarizing large volume of data, establishing and determining the magnitude of an effect, and to increase power and precision of study. Limitation. GIGO, garbage in, garbage out. Result can only be as good as original data is valid. Applies apple and orange effect, tendency to average, match together different effect. File drawer effect, publication bias relies on sheer subjectivity rather than objectivity. Result. Okay. अब हामी अर्को टपिकमा जाँदैछौँ. एभिडेन्स बेस्ड मेडिसिन प्र्याक्टिसेस इज कन्सिडर गोल्ड स्ट्यान्डर्ड फर क्लिनिकल प्र्याक्टिसेस. Aim to apply best available evidence again from scientific method to clinical decision making. Highest importance is given to strongest epidemiological studies, most important meta-analysis, systematic review, blinded